Core component mock, and this is mock number 2. Question 4. We've got a rather complicated diagram. Part of the process of protein synthesis, known as translation, occurring at organelle X. So you know in a cell with a nucleus, DNA in the nucleus, we've got transcription going on, essentially copying a gene, producing messenger RNA that goes out to the ribosomes where translation takes place and we make a polypeptide chain. So stating the name of this organelle, it's where, so whereabouts, so this will be out in the cytoplasm. So one of the things you should definitely know, translation occurs in the cytoplasm. Name organelle X, again, translation on ribosomes. So that's fairly straightforward. And the chemical components that it's made out of, chemical. So we want names of chemicals. And what a ribosome is made out of? Well, one you should definitely know, which is rRNA, or ribosomal RNA. And the other stuff is protein. If you're ever not quite sure about what anything living is made out of, protein is usually a good suggestion, as most things are made out of some sort of protein. So you should definitely have got cytoplasm, ribosome and RNA, certainly. 3 out of those 4. Name molecules A and B. Again, reasonably straightforward. A is this cloverleaf shaped molecule. B is a nucleic acid, a single strand of nucleic acid that came from the nucleus. So these should be fairly straightforward. We've got tRNA, or transfer RNA. Transfer RNA. Be careful with that, it's not transport or anything, it's transfer, because it's doing some transferring as its job. And mRNA, which is our messenger RNA, so it's taking the gene or the message to the ribosomes. Describe how 2 amino acids are joined together. Again, this should be fairly straightforward. If I draw an amino acid, it's a 2-carbon chain with one end, it's got a double bond O and OH, so that's the acid end, the carboxylic acid, and the other end it's got the amino end. Middle carbon has got four bonds. One of them is an R group and one of them is a H. And then if we draw that again here, so 2-carbon chain, the Q group, carboxylic acid, the amine group, the R and the H, and how they're joined together, well, we're just going to eliminate water in a condensation reaction and then join up what will be left. So what will be left will be, if I draw the same thing on each side there, and we have got rid of the water, so we can just join those up, and the name of the bond will be a peptide bond. So name the bond, peptide bond. Let's name these 2 ends that join together. So we've got the amine The amine group links to the carboxylic or carboxyl group. We are eliminating water in a condensation reaction. And in fact, in the mark scheme it says if you'd put these on a label diagram, that would have been acceptable as well. So you should definitely have got 3 marks there. So far, things that we have definitely got, should have definitely got cytoplasm, ribosome, rRNA, tRNA, mRNA, straightforward stuff, and these 3 marks for this straightforward making a peptide bond. It just got a little bit more difficult from here on. One other molecule not shown in the diagram. Well, for this to take place, for us to make things usually takes an enzyme and some energy to do it. So we can give either of those 2 suggestions. So for example, ATP is needed to build things, and also an enzyme to put things together. But we might not know the name of the enzyme, but we certainly know the name of the ATP, so we're going to go for that. A random change can occur in the sequence of DNA bases which occur for a protein, and the general name for this type of change, that is a mutation. And you might not know a lot about mutations, but it's using your knowledge of protein synthesis and the structure of proteins to get these next 2 marks. Explain how the change in the base sequence of DNA causes a genetic disease. So we've got different base sequence, and then that will be so different codon on messenger RNA. So a different triplet base sequence in DNA will give a different triplet for our codon on messenger RNA going out into the cytoplasm. And then here we go, you can use the diagram here. So if you've got a different codon there, we would have a different anticodon on tRNA, and so that would bring a different amino acid, and so this chain would have a different amino acid in, which could be crucial. So a different anticodon on a different tRNA, so a different amino acid, possibly a different amino acid, might be the same amino acid because of the degenerate code. We won't go into that now. A different amino acid, and so a different protein might be primary, it might affect the secondary, it might affect the tertiary structure, and so you get a different function. So and your 2 marks could have been anywhere in there. And then give an example of a disease you might not know many of these. Just your general knowledge here. The sort of typical examples are cystic fibrosis. It's just some very, very small mutations on one gene. Sickle cell anemia, if I can spell that right. That's just one mutation, 1 point mutation that leads to one change in one amino acid, change between glutamic acid and valine, and that has a big effect on sufferers of that. We could have also had hemophilia, and lots of other suggestions there. So 14 marks seems quite difficult, but there are quite a lot of straightforward marks in there as well.